Okay. You see the screen? Okay.
Yes, Prof.
Yes, Doctor.
Yeah. Okay. I'm going to use this one. I'm going to use this one. Okay. Okay, guys. Because it is not. I'm not that comfortable with what we call this online lecture like this because I need to be more interactive because virology is not the. It's easier for us to be interactive. But anyway, just to let you know about this course, I'm going to be with you for the next, I think, maybe six lectures or 7 lectures. I'm not sure. I forgot. I need to check the schedule back. Um, but basically, attendance for this course, because this is actually online, it is not that difficult. But I'm going to jot down the name appear on the screen, and you need to show your face. Make sure that you are active. I don't want you just on the video and then you disappear. That's what the office asked me to do it. To me, I don't mind if you don't come. That's your problem. But since that is a requirement from the coordinator of this course, then I need to do it. Sorry, lah, guys. Okay. So that is that one. And another lecturer, maybe they give you a quiz for each lecture. Another lecturer will give you a quiz for each lecture to make sure that you stay awake during their lectures or during their online sessions. So I don't think I'm going to be that stringent to give you a quiz, but I need you all to pay attention to this. And then I hope that you all can make references. You can check no internet, YouTube, website. There are so many resources there. Okay, this actually roughly about this roughly what you need to know about virology. So what is virus? You say infectious, okay? Infectious. So infect what? Virus infect what? What does virus need? No? So the virus is not like any other any organisms. You can find your own food. Bacteria can create their own food, their own nutrient. They can do it. They can multiply themselves. But virus, no, it is not like that. No? So the virus needs cells. Virus needs cells, living cells. Okay, can be human cells, animal cells, plant cells, fish cells, bacteria cells. So bacteria are actually cells. Okay, bacteria can be affected by virus. You know that when you were in the school, you can see that bacteriophage, meaning that the virus that infecting bacteria. So in order for the virus to stay alive and replicating, they need cells. Okay, they need cells. No cells, they will die. They cannot remain in the environment for so long. They cannot remain in the environment. And normally, they can remain in the environment sometimes not if they are actually in dead animal, because dead animals still have some cells there. I mean that cells actually serve as a protectorate, and they protect the virus. Okay? So the virus actually can cause infectious diseases, and then some of the virus can cause cancer. Okay, like this papilloma virus. Cervical cancer for ladies, for girls. No, uh, cervical cancer actually caused by papilloma virus. How come virus can cause cancer? It's very easy. Cancer occurs because of the transformations of the cell itself. You know, so there is a alteration in the genetics of the cell itself. So when your cell actually, the gene of the cell actually transform or altered, so especially the one that are responsible for the uncontrolled growth, so the cell can be transformed and become cancerous. So that's why some virus can cause cancer. So the virus not directly uh, what we call this causes multiplications of the cell, just like in cancer, but it causes alterations in the genome of the cells. So the cells won't stop dividing; they become immortal. Okay, become immortal. Um, you need to know virus. You see, you need to learn to study all of this. It's very important. Look at what happened to the world right now. Unrelentless, continuously, what we call this exposure to many types of viruses. You see, last time you talking about what Nipah virus when you were younger, lah. Okay. Then after that, SARS, coronavirus, influenza is always there from many what we call this for thousands of years now. More than 2 million people died because of the Spanish flu. It's about a few hundred years ago. So, what about COVID? That Spanish flu caused by different family, virus family. Now COVID, coronavirus is a special. The coronavirus is a family name. Coronaviridae is a coronavirus is a virus from the family of Coronaviridae. Yeah? You will know later about what is a viridae, whatever. So you see, coronavirus is very versatile. Be transmitted to many people, even though they say that mortality is low, 5%, 5% mortality. That because of the medical attention. Can be some Malaysia about 2% mortality, and certain places about 5%. So mortality occur less than 5% because of the proper medical attention. What will happen if there is no proper medical attention? You get the disease, you just got the flu, no need to go to clinics. So stay in the house, bedridden. So what will happen? The mortality may increase. Okay, this is actually the issue with the situation right now. So let me ask you, what is the size of the virus in general? What is the size of coronavirus? About 120 nanometers. Okay, so viruses are the size of the virus actually ranging from maybe 300 nanometers up to 20 or 10 or 15 nanometers. The size is very different size. From very small, about 10, 15, until 300 nanometers. All viruses are nanoparticles. Anything about that, what we call size, we call it nanoparticle. If anyone, let's say here, cannot hear me, please let me know. Right? Um, in order to understand COVID, for example, you need to do experiments. If you don't do experiments, then you don't know the behavior, the characteristics of the virus. In order for you to come up with the vaccines, you must know the virus, you must understand the virus. You cannot come up with any vaccine if you don't understand the virus. What do I mean to understand the virus? Many things. The disease, not the disease itself, the characteristics of the virus itself. Okay, later on you learn. And also, the next one, vector-based virus-based vectors. Okay, what is vector? Vector is actually a carrier, meaning that vector will carry something to do something, carry some element or truth. So you can modify virus so that virus can do something for you. Okay, what kind of things? There are many things, including antigens, including immunology, including that one you learn later a lot about virus-based vector. Meaning that you can use virus for the virus to do something useful for us. Okay. So any question about that? No questions?
No.
Okay. Just to make sure you're still awake. I don't know how many. I don't really see how many of you online, but then because I not much time there. Okay, have you heard about this disease, foot and mouth disease?
And
Yes, doctor.
How many of you know about this? So this is about one of the example of disaster in 1980s, 1980s, before you were born, you know. So this is actually in the UK. How many animals been slaughtered? 4 million animals been slaughtered. You see, this is actually what happened in UK. This is what you see the effect of the virus in animals. In human, now before COVID, nobody think so much about COVID. And when COVID first started in China, back in December and January this year, December last year, January this year, even you see WHO never think that it's going to be pandemic. Oh, not yet. You know, we don't need to declare pandemic. See, and now it's actually a big, it's very much, pandemic is everywhere. So here, foot and mouth disease virus, FMD, foot and mouth disease virus. In human, we have something similar. We call it,
HFMD.
yeah, HMD. Okay, hand mouth disease, and normally occur in children.
Children.
Yeah, children. And there are lots of these cases previously, 10 years ago, 15 years ago, in Sarawak. Sarawak. So that's in human, okay? But this different. Foot and mouth disease caused by different virus from HMD human. Okay, foot and mouth disease virus, because the disease itself doesn't cause direct mortality to cattle, okay, to animals, okay, but but morbidity is very high. Low mortality, high morbidity. But why a lot of animal die? Because no animal involved with this one cannot move so much and cannot eat so much because actually the mouth actually been severely injured because of the virus application. So mortality because of the starvations, these are all secondary infection. So you see how the virus cause problems in animals as well as human. Can be direct, can be indirect. Okay. So FMD in Malaysia is one of the major issue. Malaysian government spend I think almost about 8 millions or maybe 10 million ringgits a year to vaccinate few hundred thousand cattle in Malaysia. Malaysia standing population of cattle about 500 to 600 now thousand. So we spend about 8 million to 10 million ringgit a year. I mean, our government to vaccinate animals, and then it is free. Department of Veterinary Service is doing that. Okay. And where we normally see this foot and mouth disease virus in Malaysia, and how we are getting this one, the virus from the normally from the importations of animals into Malaysia and so on. And then improper, what we call this transportation of virus from what we call this our neighboring countries. Okay, virus, no, Latin actually means poisons, poison. Okay, and then because during ancient time they don't know what is this, they just see the disease and then die, poison, and then maybe related to witchcraft, whatever that's what they think about. So when you what we call this identification of virus is related with the highly related with the immunizations. You heard about the story in the school time, no? Edward Jenner inoculated human with cowpox, and then because of the cowpox, the human developed lesions against cowpox. Cowpox actually pox virus from animal and given to human. Then human protected against smallpox. And they don't know what kind of things that they agent. That one in the 18th century, what, more than 200 years ago. So cowpox and smallpox. Smallpox, you know smallpox? Any one of you got vaccinated with smallpox? Yeah, vaccine? Yeah. Normally on the left hand side of your shoulder. You have it or not? You don't have, right? And your generation, you don't have it anymore. You can see the depression of the skin. So smallpox is actually in Malay they call it cacar. They just poke around and then see. Your right hand side normally they do TB. You can see the bulging skin. That one is tuberculosis. Two different things. So maybe during your time, you don't have smallpox vaccination because smallpox virus has been eradicated from the world about 20 years ago, more than 20 years ago. So smallpox virus still being stocked by American government as well as US government because they're afraid of the potential WMA, weapon of mass, not WMD, weapon of mass destruction. They don't want.
Doctor, Prof, I think you accidentally muted your mic.
Okay, can you hold on? To this. Can you hear me?
Yes, Prof. Yes.
Yes,
Okay.
Prof.
Uh, Where is my slide? So that's why you need to understand this virus, no? Then smallpox virus has been eradicated, but the US still come up with a stockpile for the vaccine. So smallpox vaccine is available, and then about 10 years ago becomes 10, 12 years ago becomes popular because of the potential weapon of mass destruction. In fact, Malaysian military, Ministry of Defense, actually they order um, about 2 or 3 million doses of smallpox vaccine from US. But that's a confidential at that point in time. You know that because they're afraid of the war. I mean, there are people using smallpox virus to spread this one. It is not like what you see in the TV, zombie and so on. Okay. Um, Okay, this is the story. I'm not going to go. You can read this one. Tobacco mosaic disease virus (TMDV). You can read on your. This one actually from animal from plants. Tobacco actually used to have this kind of facing a lot of losses because of the disease to be alleged. Okay. Uh, what are the characteristics of the virus? Number one, virus is a living particles. Okay. Virus is obligate parasites, meaning that they need host cells for them to replicate. So virus, when they enter the cell, one thing that they do, nothing else. They replicate, they multiply. That's what virus doing. Okay? Not like, worms, go into your body, they stay there, they do whatever they do. But virus enter the cell, their aim is nothing other than to replicate. Okay? Virus could only survive and reproduce in living cells. So the virus cannot stay outside of the cell. Can virus stay in the environment? Okay, COVID, you wear the mask, right? You wear a mask. The size of the virus is actually very small. So when you sneeze, assume that the person actually already infected by the COVID virus, sneeze. So how long the virus can stay outside? And how long? Hours? Days?
2 to 3 days.
Huh? 2 to 3 days. Where? Where? In your mask? Hmm?
Maybe on
Or
the
your
surface.
handphone? Is it because of your handphone? You hold your phone, yeah, the virus go to the handphone? Normally, virus cannot stay out long outside of the host because the environment doesn't allow that. They can be broken easily. You see? Because of what? Virus, if you expose the virus to the water, what will happen? Many viruses will explode, be broken because of this osmotic imbalance. You know, when we are, let's say, when we would like to propagate the virus in the lab, we need to use media, media that mimic our body osmotic, what we call this tension. If you use, let's say, dry water, distilled water, they will die, they will break, will be broken, just like that. So see, virus is actually it's fragile. Bacteria, yes, they can stay, can form spores. Virus cannot form spores. Bacteria, yes, they can becomes dormant to form spores. But virus, yeah, just virus. Okay. Let's say herpes virus, the one that caused AIDS. How long it can stay outside of the host? Only minutes, few minutes. Once they are exposed to UV light, finish. They are becomes inactivated. Inactivated meaning that kill, you know. Inactivation or inactivated, there is a terminology in virology. In the inactivated virus means that kill virus. Okay, understand? Inactivated virus, inactivated vaccine, whatever that is actually kill. So here, when the virus actually enter the cells, I don't know whether you understand that. Assume that this one is a petri dish, you know. This one, you look at the screen, this one is petri dish. Then you grow a cell, one layer on on the surface of petri dish, and then you add virus. Okay, the virus will grow inside that particular cells, and then the cell will die, and this is what you're going to see. The this whole meaning that the cell already died, no more cells there, they died. And this dark area, there are still cells there, survive that. So this is what we call area that caused by the lysis of the cell because of the virus replication. Doesn't matter whether the whole layer is actually animal cells or bacterial cells. So this kind of thing we call terminology. This one you're going to see this one later. Plaques, PLAQ, no PLAGUE, plaque. Okay, that's terminology you're going to see in practical class later on. So this formation, this actually the empty area here, whole area, you can count 1, 2, 3, 4, 5, 6, 7, whatever. The more virus that you have, the more hole that you have. So this become later on you become a technique for you to know whether the sample contains a lot of virus or less virus. If it is a lot, how much? If it is less, how much? How many? So this that is the name of the test. Now later on you learn. Right. No need to go for this cabinet pictures. Okay. What virus actually obligate intracellular parasites? Bacteria, bacteria actually a parasite. What is parasite? Parasite is the one that actually invade the host, stay there. They are not making their own food. They just conquer and take over whatever we have. So this is what we call obligate intracellular parasite. Obligate means must. Without cells, they cannot be there. So virus is very simple. Very simple. So virus actually contain only our cells. We are multicellular organism, fish, animals, worms, no? Even bacteria. All of them, they have DNA and RNA, both. They have DNA. These two types of nucleic acid, they have it. DNA and RNA, they have both. But virus only have one of these, either DNA or RNA. No virus have both DNA and RNA together. They have one. So that's why people will ask, COVID, is COVID DNA virus or COVID is RNA virus? RNA or DNA? So what is so important about the difference between DNA virus and RNA virus? Okay, very important for classification, mutation, transformation, disease, and so on. So DNA virus is more stable than RNA. Remember that. So RNA virus, the rate of mutation is higher than DNA. You will learn. Don't no need to worry so much about the information. And you see, the one that cause disease out there, outbreak. You see, what kind of virus so far? Flu virus. They are RNA virus, right? MERS, RNA virus, right? MERS, SARS, RNA virus. Okay. Newcastle disease virus. Have you heard about this virus before? They are RNA virus. Yeah. So, coronavirus. They are RNA virus. Corona. What is corona? Crown. Corona means crown. Yeah, crown. You see when there is eclipse, no? You see Grahana. What can you see? So when you see the virus under microscope, looks like that. Corona, crown-like. Okay. So the name of the virus is quite interesting too. So extremely genetic simplicity contain DNA, RNA. Human, how many genes that we have? Our genome encoded about 30,000 genes. Okay. Virus, some of them may be 100, some of them may be 5 or 7. Small virus, very few genes, but they are infectious. Some of the bigger virus, like pox virus, they have many, but still less compared to human. 30,000. They may have maybe 100. That is most, no, at most. Okay, parasite. How they replicate? They produce eggs. They have what we call this sexual or asexual, what we call this replication. Just like cells. Cell actually they have binary fission, right? They split into 2, 4, 8, 16, and so on. But virus doesn't work like that. The virus use our cells, use animal cells, use a cell, the host cell, as good as those cells as a factory machines, just like a factory. If you want to produce a car, you have a car factory. What are you doing there? You're going to bring tire there, you're going to bring doors there, you're going to bring whatever there. Then after that, you pack together, you assemble together, right? So that's what virus doing in our cells. The virus enter the host cells, so they take the nucleic acids, they take the protein, they form their own shape, then they come up with the virus. They call it packaging. They call it whatever. So that's why virus replication involve disassembly and assembly. Okay, assemble and replication by one step growth. This one I will explain later what is one step growth. So replication involves with disassemble. When the virus enter, they remove their protein whatever. So they put their virus nucleic acid into the cell. Virus nucleic acids take over the host cells. Whatever instruction, the code, whatever they do it, the virus take over, and the virus give instruction to the host cell to do things for the virus. That's how the virus. So the virus invade the host cell, and then they take control of the cellular mechanism inside to produce more virus. That is okay. That's only job of the virus. Enter the cell, take over. Okay, you produce parts, everything for me only. For the whole cell, very minimum. Allow the whole cell to survive for some time before the whole cell die. Okay, that's the virus. You can look at that kind of imagination. Okay, so the difference between viruses and cellular microorganism. So viruses are viruses. Cellular microorganism, including animals, out to the bacteria. Bacteria are cellular. Okay, growth. Virus grow only within the cells, obligate. But cellular microorganism, free living. What does it mean free living? They can be on their own to replicate, to grow, to live, to get the whatever nutrient and so on. Viruses cannot do that and won't do that. So nucleic acid contains DNA or RNA, but other cellular always both DNA and RNA. Okay. So when you have DNA or RNA, what are you doing? You go to the lab, you can check, hey, this virus, is it RNA virus or DNA virus? You can check there already, it's a part of the diagnostic method. If it is RNA virus, these are the list of RNA virus. If it is DNA virus, these are the list of DNA virus. What do you want to do with it? To grow them, require different medium, different cells. Enzyme content, very few if there is any enzyme. Most of the virus does not carry any enzyme inside the virus particle, except for few viruses, big viruses. What are those viruses? Pox virus carry enzymes, 1 or 2 enzymes, and this retrovirus, I mean the virus that can cause AIDS, no? They carry enzymes, okay? But cellular microorganisms have many kind of enzymes, So cell component, of course, no, they are not cellular. They don't have ribosome. They have no enzyme. They have no mitochondria. They cannot generate their own energy. They cannot do anything. See viruses. But cell components, no, cellular organisms, of course, they have ribosome, enzyme, to generate energy, to synthesize protein, whatever, they can do that. Yeah, they cannot do anything. So that is the situation. Okay, see, this what is the size of the smallest virus? About 20 nanometer. The largest maybe 300 nanometers. Smallest virus, largest virus. COVID in between the two. Coronavirus between smallest, coronavirus about 120 nanometer. So this largest virus is, we're talking about pox viruses. Okay. So genome content, 4 to 5 genes only. Human, 30,000 genes, more than that, maybe. And animals, different animals also they have that. So this small number of genes can cause havoc in our life, can cause many diseases. Largest virus, about up to 100 or 200, normally about this. So bacterial cell, E. coli, 3,000 genes. More bacteria already 3,000 genes. Human cells, about that. Okay. And then, do you understand this or not? Transcription, translation. What is it? What is a gene transcription, translation? What?
Transcription is like you convert the DNA into mRNA.
It's not conversion. There's no such thing conversion. They actually they produce mRNA based on the template, based on the coding system. Okay, there's a word, but you can say convert, but the actual thing is no conversion. So transcription is, sorry, it's a spelling error. Transcription translation is actually a mode for productions of the protein. Transcription, mRNA, then after that, you get the translation to produce protein. So this is what virus doing, transcription translation. And the other one is, I put here, is actually nucleic acid synthesis or DNA synthesis or RNA synthesis. There are only two pathways, One is actually to produce new nucleic acids for the virus. The other one is to produce new protein for the virus. So that's what the virus is doing when the virus infects the cell. The aims, the major aims of the virus actually to do this, to replicate. So to replicate, they need to produce nucleic acids, DNA or RNA, and they also to produce protein. These are actually two different big pathways. And they produce protein, they produce nucleic acid, then they assemble. They do packaging, really assemble, just like a factory. I mean, it's amazing, they produce protein first, then you can see the nucleic acid go into the protein, So this is actually the functions of the host enzyme so that the virus form. It's amazing. So virus is not cell, so this actually violates, the original theory of living and non-living. Is virus living or virus non-living? Confused. In fact, of course it's living, but it is actually non-cellular. Some people disagree with that, but that is a fact. So from time to time, you will discover this terminology, virion. What is virion? What is virus? So virus is actually general idea about virus. So virion, normally we refer to the single virus particle. Virion, single virus particle. And sometimes we refer this one to the new virus that produced by the cells infected by the virus, infected cells. So new virus, we also call it virion. Okay. And one thing, these durians, no? Durians. So the shape of the virus, you can imagine just like a durian. Okay. Inside that durian, you have the DNA or RNA. Outside, you have protein, we call it capsid. So the skin of the durian, you can imagine that as capsid. Inside that, you have DNA, RNA, capsid. So that is a basic structure of the virus. So inside, you can imagine that inside the ball, the durian, the fruit there, they have a DNA, RNA, and then the one that coating the durian, the skin itself has capsid. Okay. So what about the thorn of durian? The thorn. That virus also have a structure like that, something like that. We call it spikes. Spikes. Durian spikes. Spikes, thorn anyway. Spikes, or maybe certain old terminology they call it papilloma. You don't worry about terminology. You learn that. It's in the slides. So capsid actually very important for the interaction with the host cells. Capsid. So capsids must interact with the host cell. If there is no interaction between capsid with the host cell, the virus cannot enter the host cell. Okay? So that's why not all virus can infect disease. Chicken virus, Newcastle disease virus, cannot infect cattle. Why? Because capsid doesn't have the necessary protein that can interact with the cattle cells, a cell from buffalo, a cell from what we call this from cow. They don't have it, so that's why that virus cannot infect cattle cells. Okay, all right. So in capsids, capsid together with capsid, they have additional layers: carbohydrate, lipids, other protein. Don't have about this. So any questions? Yeah. No questions? No sleep? Understand, Aaron?
Yes.
Yes, doctor.
Yes, doctor.
Okay, crystal
Yes,
clear, okay?
doctor.
Okay,
Yes,
no,
doctor.
no coffee on your table, right? All right. So, as I said, as I mentioned just now, it's either DNA or RNA. This is now you go DNA virus or RNA virus. Classification already, composition. So, DNA virus also they have double-stranded or single-stranded. Can be single-stranded DNA, single-stranded RNA, or double-stranded DNA, double-stranded RNA. What does it mean? What does it mean?
The structure?
No, you remember about DNA, one single thread line. You know what is the code, DNA code? A, T, C, G, G, C, C, A, whatever, right? There are only 4 codes, right? A, T, C, G. Adenine, thymine, huh? Cytosine, and
Guanine.
A, T, C, G. Remember? No, basically, we are encoded by these 4 different nucleic acids, A, T, C, G. Okay? You know that? So if it is single stranded, you only have A, T, C, G, G, C, C, whatever. One line, they are actually arranged in sequence. But if it is double stranded, you can have pairing A with T, C with G. Okay? They have double stranded there. In the class, you need to show that, double stranded. A, T, C, G. So you try to understand what is double stranded, what is single stranded. And why some of them single stranded, why some of them double-stranded. I wouldn't come out with that kind of question in the exam. Why these viruses have double-stranded, not single-stranded? The answer is I don't know, because that is what the nature, given to us. You see, right? We don't know why this one double-stranded. Why not a single-stranded? I cannot answer that question. Nobody can answer that question. Okay, and they also have positive or negative sense. Sense. The positive sense and negative sense. What is positive sense? What is negative sense? Positive sense and negative sense. They are related with the transcription translation. Okay? If positive sense, meaning that the viral genome that itself, the viral genome itself, single-stranded or double-stranded, they themselves can act as a transcription factor. They can do direct the protein synthesis. They act as mRNA. That's what positive sense. If it is negative sense, means that they become a mirror image, a template to produce another DNA RNA. Okay, meaning that they becomes what we call this a block for them to produce a positive strand of DNA, mRNA. So meaning that positive strand of DNA, the associated strands of the DNA RNA act as mRNA. But the negative one, they have to use that to produce the mRNA, a mirror image. You don't understand about this. This is about nucleic acids, biochemistry nucleic acids. You can ask me anything, anytime. And then nucleic acid, can be segmented or non-segmented. Segmented means that inside the durian itself, there are many segments. Many line. 1, 2, 3, 4. Non-segment, we only have one continuous strand of the DNA or RNA. One single strand. Just like a cable, like that, one single strand. That's one, we call it non-segmented. Segmented means that one strand, two strand, three strand. Each strand there, they carry different genes. They no repeats. They don't repeat. No? They don't have copies of several copies of the segment in the same what we call this virus particles. And also geometry, the shape of capsid. This one can be helical. Helical means helix. just like what we call this electrical cable. You can imagine the electric cable. Out there, you can see the what we call this coated by rubber. Inside, you can see the coil. The copper coil. So inside the copper coil, then it is that one is maybe DNA. The one that surrounded the what we call is the copper cable. We call it capsid. That's how they come up. It's not, instead of durian shape, it becomes a filament shape. That's how they call it capsid, helical concepts of the nucleocapsid. So icosahedral is actually diamond shape, of the nucleocapsid. And also the next one, virus, some of them have a lipid bilayer, and some of them doesn't have lipid bilayer. Okay. Capsids. Remember durians? I mentioned to you. There is many viruses. They have another layer outside of the durian shape, and that layer we call it envelope. So in principle, you imagine that the round figure in the core, in the center of the durian, you have DNA or RNA. Then DNA or RNA is surrounded by capsid, and capsid surrounded by envelope. Understand? You have that two layers. So envelope is actually from cells, in the cell membrane, the infected cell membrane. Don't worry so much. You cannot remember all of this. It's okay. During lectures later on, I will repeat this because this is important for you to understand the concepts of the virus. So this is actually nucleic acid, adenine, guanosine, thymine, and cytosines. You can read on your own on this. So if it is DNA, you have A, G, T, C. Okay. If it is RNA, you replace T with uracil. Okay, RNA only have AUCG, not ATCG. Okay. What is structural protein and non-structural protein? You learn biochemistry, right? So structural protein, that protein with form the structure of the cells of virus itself. Non-structural, what are those? Basically, they are enzymes. Enzyme actually non-structural. So what are the functions of non-structural protein? They act as to help, something becomes a factors. This actually the protein that act as a to catalyze, the formations of certain protein, viral protein, or they act as an enzyme and so on. So that is a different issue about the non-structure. Now we talk about the structure protein here. So what are the function of the structure proteins? Capsid, it is a structural protein. Envelope is actually a structural protein. And on envelope itself, there are several different types of structural protein on envelope. Okay? Capsid also, they have several different type of what we call this structural protein there. So what is the function of the protein? To protect nucleic acids. Nucleic acids is very fragile. You expose to UV light, they cross-link, and then they are not active anymore. So this actually protein also attached to the cell receptor. If there is no protein, viral protein cannot attach the receptor on the cell, then cell won't be affected, then you won't get infection. So that's why some virus can infect only certain cells, not every cell. Some virus, many chicken virus cannot infect humans because of that virus doesn't have these receptors on our cells. No receptors, for that virus. So it's unique. So protein also important for the virus to penetrate cell membrane. They must go into the cell. They have to penetrate the cell membrane. Okay? Protein also important for the virus replication, nucleic acid replication. And what we call this protein also important for the initiations of the virus replication. Must, for the virus to start, they need something to be triggered, They need certain protein for them to be triggered. And this actually the protein useful for the virus replication can come from the virus itself or from the host. And many cases, viruses, when they enter the host, they modify host cell, including the papilloma virus, pox virus, so that the host cell becomes suitable for them. Okay? So non-structural here, just mostly you can think of enzymes. Enzymes are non-structural. Okay, let's see this one. So this is actually the shapes of the one of the virus. We call it herpes virus. You know herpes virus? Have you heard about herpes simplex virus in humans? Herpes virus. Herpes virus is actually DNA virus. It's a big virus. Okay. So here, inside here, looks like durian, right? Inside here. You see the shape here. If you look carefully here, so this is actually the shape. You see the shape, and some sort of pentagonal here, but this is not really pentagonal. This is actually the shape of the virus captured under electron microscope. This is what we call icosahedral shape of capsid. This whole thing are the capsids. Okay, so this the whole thing here it actually is an envelope. So this is actually envelope. So there is a space between envelope and the capsid. So this space contains certain protein, normally non-structural, becomes a factor for the virus to start the replication when the virus infects itself. So in case of herpes virus, we call it tegumen. So this is actually herpes virus, and herpes virus is envelope virus. Okay? Right? Envelope, yeah. So, inside here, can you see that dot here? So each one is actually a square here. You can see that this is quite clear. Each one here, we'll call it capsomeres. Capsomeres, many capsomeres becomes capsids. So capsomeres actually individual units of capsid. Okay, capsomeres, each protein blocks here. So the whole thing become capsid. So nucleocapsid is actually because of the nucleic acid, stand for nucleic acid, and capsid because of the. This is sometimes when we call it nucleocapsid, is a terminology to indicate that nucleic acids together with capsids. Okay. Okay. This is what you see in the herpes virus just now. Icosahedral. Naked icosahedral means that no envelope. Okay. Naked icosahedral meaning that no envelope. There is supposed to be envelope outside here. So this one naked like helical, no envelope outside. This is actually, and this is actually the capsid, the one that I just mentioned. I mean something similar with the electrical cable here. Okay, so this is actually icosahedral shape. Icosahedron. So I don't go into detail. Just want to show you diamond. Diamond looks like diamond shape, but it is not really diamond. But this is how it means what we call this. Then been named. Okay. All right. Now we're talking about virus. Now you know about virus already. There are many types of viruses actually. How many viruses that we know? There are many virus families. Virus family already what 16, 18, 20 different virus families. And I give you an example. Herpes virus itself. There are few hundreds herpes virus, different herpes virus. Just herpes virus already. few hundred. So how many viruses that we know? Thousands. So you want to learn virology on the 1,000 different viruses? I can't. I myself cannot remember everything. Impossible. Even though you get 10 what we call this, you graduate with 10 DVM degrees, you wouldn't finish study this. Okay, it's so many. So there are 4 different morphologies of virus. Icosahedral, you have seen that just now. Helical, cable-like structure, envelope, and complex. So envelope virus, complex means that is normally actually related with the big virus. Okay. So just now I mentioned about capsids, capsomeres. Many capsomeres together form capsids. Okay. And this one, I think you better know lah, no need lah, self-assembly, no exam. This is about the later stage of the virus application. Maybe not applicable right now. So you see, this is actually big virus. This is more towards complex. You cannot say icosahedral, you cannot say helical. Maybe you can say envelope, but it's complex structure. Okay. This round structure. This is related with what we call this influenza virus now. This is a bacteriophage. Okay, bacteriophage. This is not clear. This is actually pseudo-rabies virus, bullet-like shape. Long, slender. You see the round end here. This is actually rabies. This is a rabies virus. The one that you have an outbreak in the Sarawak. This is a virus, rabies virus. This is a bacteriophage. Okay. This is adenovirus. Okay. This is more towards this herpes, this pox, this pox virus. This is actually the. This is about the maybe influenza virus here. It's not clear this one. Okay. So adenovirus here you can see, and you can see the something what we call this come out here, papilloma here. So this is a papilloma virus. Papilloma virus. You can see capsid. This is a parvovirus. Parvovirus cause diarrhea in animals. Okay. This is a morbillivirus filament type. Okay. And this the size of the virus. So adenovirus, adeno. What is adeno? Adeno means gland. You learn that later just to expose to you. Adeno means gland, meaning that the virus infecting glands. Papilloma is the one wart, the one that causes wart. Another, this one, the herpes virus here. You have seen this before. This influenza virus, para-influenza, you see segmented, different length, different copies. This influenza virus, and this actually pox virus, complex virus. So icosahedral shape, pleomorphic. What is pleomorphic? Pleomorphic means that morphic is actually a shape. Okay, pleo means it is not set. The shape is not set just like this one. So here can be round, can be long, egg-like, can be this shape. Pox virus, they are quite consistent. The shape under microscope you can see like this. They call it brick-like shape. Even like brick to me, it's more towards pineapple kind of look. But this is what they describe, in the textbook, brick-like shape. So it's a phage virus, adenovirus, influenza, potato virus, whatever. Okay. How to move forward? This is right. just to show you this rabies virus. Okay, you can see that the name here. Rhabdo here. Rhabdo, rabies here. So that's how rhabdo name come from. Okay, bullet shaped virion is the size. You can see the envelope. This is actually envelope here with spikes, protein like this. Okay, there is another layer here, protein here. This one. This is what we call matrix protein. You just name matrix protein. Many viruses have this one, another layer under envelope, a part of the envelope. And then helical capsid. Yeah, you can see that is helical. And genome have one single stranded RNA, 13 to 16 kilobase of only they have only 5 genes. Kilobase means that 16,000 ATCG and so on. So it is not a big virus. Okay, this is actually something that you need to know about this enzyme. Huh? For virus require enzyme for them to replicate. This one, you know. Reverse transcriptase. Reverse, you know the word reverse here. Transcriptase. Transcriptions. Okay, transcription. For HIV, HIV actually is an RNA virus. But they need to produce DNA for them to produce protein later on. Okay, protein. So don't ask me why they need to do this. Other viruses that not to do this. I also because the nature is like that. Okay? It's characteristics of the virus itself. So they carry this enzyme to convert RNA to DNA, so for them to produce a protein, and also for them to replicate. They replicate, no? They produce DNA. Then after that, they reverse back to RNA, so become virus. So these are the enzyme, important enzyme for you to know. Polymerase, polymer to build a new nucleic acid. In that they try to polymerize, no, to make as many new DNA or RNA as possible. So you know this COVID-19, the test they use, they call it PCR. You know this? Polymerase chain reaction. Polymerase chain reaction. So they use this enzyme in the test to produce as many new, what we call this, nucleic acid as possible until the machine can check. Yes, this is actually the COVID-19 RNA virus. So this is actually the test that they use, polymerase chain reaction. So maybe the sample contains 2 or 3 copies, maybe 10 copies of the virus. You do the normal test, you cannot detect the virus because normal test low sensitivity. So they use this enzyme to make it as many as possible, million or billion copies, then the machine can tell, yes, this is actually the nucleic acid of the virus. And also enzyme that can help the virus not release. Yeah? Have you heard about this one? One. And one. Have you heard about this? Huh? Flu virus, H1N1, H1N5.
Doctor.
You read that from newspaper as well, right? So,
Yes.
and here, this one, Uramidase. It's not only for Newcastle, also for flu virus. Also for flu. Okay. Neuraminidase. H stand for hemagglutinin. It's also another protein enzyme. Hemagglutinin neuraminidase. So neuraminidase actually on the virus itself that determine the type of the what we call this a classification of virus. Lysozyme. This one is actually important for the destructions of the cell so that the cell will release the virus. Okay, any question? Any question there? So basically this, okay, two phases of virus life cycle. One is actually as a particle, the other one is replication. These are only two. So if it is actually as a particle, they're present in outside of the cell. We call it extracellular space because not our tissues, no, inside our body. So between cells, there is a space. This is what we call the extracellular space. Okay. The virus contain nucleic acid and protein code, and also perhaps protein layers. Contain nucleic acids, protein code. Nucleic acid, DNA, RNA. Then after that, you have capsid. Some viruses have nucleic acid and capsid. Some viruses have nucleic acid, capsid, and envelope. Okay. So the virus with capsid, the virus with envelope, we call it envelope virus. The virus without envelope, we call it naked virus. Non-envelope virus or naked virus. So replication always intracellular. They actually take over the host metabolic machinery pathways to produce new virus for replication. And then they exist mainly as replicating nucleic acid, DNA, or RNA. Inside the cells, it is what you're going to see in many cases. Okay. COVID, how long it takes for you to get the disease from the first time you get infection? 14 days, right? So what happened between day one or day 1 minute, the beginning of the day one, until to day 14? What happened? The virus replicate first inside our body, cause damage or destruction to a certain organ, whatever system. Then from there you see the clinical disease, fever and so on, 14 days. So what is the virus doing during that 14 days? When the virus, first day, second day, when the virus enter the cells, the virus infect the cells, virus enter the cell, you cannot detect any virus particles at all within, let's say, first 24 hours, first 48 hours, first 36 hours, depending on that virus. Why? What happened? When the virus enter the cell, there is no more virus particle. So the virus enter the cell means that they already put the DNA or RNA of the virus into the cell, inside the cell. You cannot see virus particles because they are just about to produce new DNA or new RNA and viral protein. No assembly yet. You will only see the virus particles inside the cell under microscope whenever they complete the first cycle of the replication. Okay? Some of the virus complete the cycle within, let's say, 2 to 3 days. Some of the virus complete the cycle to produce new virus within weeks. It depends on the type of the virus. Pox virus take longer. Herpes virus, alpha herpes virus, within, let's say, 24 hours, they already complete one cycle. But gamma herpes virus, maybe 1 week. So it depends on the type of virus. Okay, understand that? Any question there? Too much for you? For the
Prof.
first day?
Oh, sorry.
Yeah?
I have a question.
Yeah, I need question as much as possible.
Okay, Prof, you mentioned that the virus particles, it can be seen under microscope after it has completed one cycle, right? Replication can. Okay, so is within 2 to 3 days, like, is there any way to disrupt the cycle?
Yes, and so are you talking about antiviral agents here? For you to come out with, let's say, antiviral, you know this, okay, you know this, because we haven't complete lecture, lecture one. Virus, bacteria, they are sensitive to antibiotic, right? Penicillin, streptomycin, whatever, or mycin. Bacteria will die because of this antibiotic, right? What about the virus? Antibiotic cannot do anything on virus. No virus are sensitive to the antibiotic. Why? Why? Because antibiotic will directly interfere with the biology of the bacteria itself, right? So what antibiotic does to the bacteria? You learn, you go home, you learn. But for the virus, you cannot use antibiotic, but you can use antiviral agents. So antiviral agents mostly are chemicals. They are chemicals. So, right. In order for you to stop from virus to complete their cycle, you need to understand the virus replication. Let me tell you, you need to do some immunization. Remember, the virus, this is actually the step of virus replication. The virus, first of all, must attach to the cell, right? The virus must attach to the cell membrane of the target cell. If the virus cannot attach to the cell membrane, what will happen? Can virus infect the cell? Cannot, right? Can you block the virus from attach to the cell? Yes. What kind of thing that we use to block the virus from attach to the cell? Antibody. You heard about antibody, right? Antibody will bind the virus. If you have the antibody to bind the virus, if you have COVID-19 virus in your body, you already vaccinated. I assume that you already vaccinated with COVID-19 vaccine. The body produce antibody. Then after that, come virus no for you. The virus enter your blood circulation. Then here antibody attached to the virus. Then the virus cannot enter the cells. Okay. The first step is actually attachment, virus attachment to the cell. That is the first step. You can block that, yes. Virus cannot replicate. Assume that the virus can attach the cell. Next step is penetration. You have to go inside the cell. Is there anything that we can do to block the virus from going into the cell? Yes, there are factors. There are factors, but not for the COVID-19. Okay, next step. The virus attach to the cell, then after that, the virus must penetrate the cell plasma membrane. Cell membrane or plasma membrane. They penetrate. Assume that they already penetrate, they are inside. Once they are inside the cytoplasm of the infected cell, what happens? The virus, once they're inside the cytoplasm, they are no more in the forms of virus particle. They are already in the forms of nucleic acids. Once they enter, virus enter the cell, they are already in the form of nucleic acid, DNA or RNA. So what happened to the capsid envelope? So they becomes a part of the infected cell plasma membrane. So virus attached to the plasma membrane. So the virus envelope becomes a part of the plasma membrane. Okay? Then they enter inside. If they have capsid will be removed, and then you have nucleic acid. So inside the cytoplasm, what you get only the nucleic acids. That is the second step. We call it uncoating. That's the terminology is uncoating. You learn that in that lecture. Don't worry. Uncoating, meaning that the capsid will be removed from the virus particle to release the DNA or RNA. So the next step is actually you have two pathways. One is to synthesize new DNA or new RNA. That's number one. And the other step is actually the same time to synthesize new proteins for the virus. You can imagine that. Two pathways there. One to produce new DNA or RNA. The other pathway is to produce proteins for the virus. Okay? If you can block the synthesis of the new RNA, that you can do that as well. If you can block the synthesis of the DNA protein, you can do that as well. There are chemicals to do that. And you can use chemical to do that. The next step is once you have DNA or RNA, or you already have protein, you want to assemble them together, becomes a new virus particle. So the assembly of the virus occur at different places in the cell, depending on the type of virus. Occur in the nucleus. Some virus prefer in cytoplasm. Where is cytoplasm? Can be mitochondria, can be Golgi apparatus. There are lots of things that happen. So each step there, you need to understand as virologists. Once you understand, you can produce chemicals or a protein to block the virus. Okay? You can use one type to tackle only a single step of the virus. Normally, it's not that effective. Normally, we target the different what we call the stage of virus infection. You can target to block attachment. You can target to block from the virus assembly. Okay, so for the treatments of the HIV patient, they produce 2 to 3 different chemicals, antiviral agents, to block reverse transcriptions. They to block the assembly. These are the things. So there are many what we call this many things that you need to understand. You need, in order for you to come up with vaccine and what we call this antiviral agents, you need to understand how the virus replicates. Different virus replicates differently. So if you understand that, you can block them from replicating. Okay. Any more questions? I hope that will answer your questions.
Okay, thank you so much, Prof.
No problem. Anything else? Okay, if there's nothing else, give me about 10 minutes. Then I'll come up with the practical, what we call this session here. I need to tell you what is sterile techniques, all about how to grow virus in general. Okay. So what is the topic of our practical today? Based on the schedule, sterile techniques, right?
Prof, we don't have the class schedule yet.
No, what do you mean? You don't have that? Oh, coordinator haven't given you yet?
No, not yet.
Oh my God, I think I need, okay, I'll get from her, then I'll pass you. So the practical class, no schedule yet? What do we?
No, not yet, Prof, not yet.
Yeah, so I think I'll try to get that. So our class is Wednesday, right?
Yes, Prof, Wednesday morning.
Yeah, I think I will pass you that. I will email to you.
Okay, Prof.
So today is basically about sterile techniques. Sterile techniques. Okay. But anyway, sterile techniques, I would like to make you actually familiar with virology lab environment, what we're doing there in general. Maybe it is a bit different from what they given you before, but I think you will learn faster. So virology, it is interesting, but there are lots of things you need to what we call this memorize, because this is about facts. Okay, it's less on the strategy, it's about facts. The key to virology is, number one, to understand the virus replication, how the virus replicates. Different viruses replicate different way as virologists. Second, if you learn, let's say after a few times you learn about this, you will get saturated about how the virus replicate. By the way, when the times, during exam, the way that we give you the questions, the answer that we demand from you is a bit generic, because you're not going to be virologist anyway. You understand that? We need you to understand how virus replicates and relate this to the disease, and then how you can get, let's say, the animals, protected from the viral disease. And then this related with your immunology. You are taking immunology class this semester, right? Right? Yeah, are you taking immunology courses this semester or next semester?
This is
Semester, right? So, okay, I think take rest about, let's say, about 10 minutes, then come back, then I think by 5 you can go already. Okay?
Okay, Prof.
Ada.
I will cut this off, then 10 minutes later I'll come back. Right.